Ask a language model for a drug target, and it will always answer. Here, it names DHOD, hey, confident, fluent, and completely ungrounded. Nothing checked the biology, no essentiality gate, no permutation control, no provenance. FluxForge is a fail-closed MCP. It integrates literature, regulatory, and protein interaction evidence into genome-scale metabolic models to predict CRISPR gene dependencies, cross-organism, and permutation controlled. Integration is the core idea. Metabolism is the best curated network in biology, so the metabolic model is the cornerstone. Every other layer integrates onto it. Regulatory networks, physical interactions scored by structure, and literature extraction all attach here. 18 type tools move that evidence, and everything crossing a gate carries its provenance. UNNSI Conagate. Here reasons The gates live in code, not in the prompt. Is there real essentiality truth to test against? Do the benchmark controls hold? Enough positives, and a lift that beats a shuffled null? And critically, does this evidence actually add value? Here, a generic literature network doesn't. Permutation null, p equals 0.587. So augment compare halts. The DAG gates on biology. The benchmark controls the statistics. A single halt is terminal. The model is physically prevented from proposing a target. Every prediction is inspectable. This is the SARS-CoV-2 host network in Calu3 cells, a viral protein, ORF10, wired to host enzymes and the metabolic reactions they control. Each edge keeps its evidence. This interaction scored an IPTM of 0.522, classified physical, matched by Boltz2. Eningcore Brichtasaltinic in create, India tree cells originally. Start with the corroboration. A human-curated network reproduces the rescue, T-Rust, hand-curated, not from any language model, lifts weak glioma baselines by nearly 9% at p equals 0.005. It's independent of the literature layer, and it kills the objection that a model simply memorized essentiality, which sets up the headline, a negative result, diagnosed and fixed. A generic literature network was a null, no effect in glioma, no effect in melanoma. Retarget the same extraction onto regulators of metabolic genes, and the lift appears, plus 0.04 in glioma, up to plus 0.04 in glioma, up to plus 0.06 in GBM and melanoma, everyone at p equals 0.05. Same engine, only the target space changed, controlled with essentiality blind extraction and a feature shuffle permutation test. And when the evidence doesn't touch metabolism, we say so. The sparse pathogen layers report exactly zero effect, p equals 1, and the permutation test agrees. That's the rescue law. Integration rescues weak metabolic baselines and leaves strong ones flat. Benefit is predictable, not lucky. And it's public. One command from pipe E, pip install FluxForge MCP, pulls every dependency and the bundle data. In Claude Code, add the marketplace and install. In Claude Desktop or Cursor, it's a few lines of config. Self-test confirms all 18 tools. 16 systems, real CRISPR and depth-map truth, every reported lift permutation or bootstrap controlled, a fail-closed MCP for CRISPR target identification and drug repurposing that integrates on a curated metabolic core and stays honest about its nulls. FluxForge by Ali Kishke AGD.